So, hello and welcome back, dear all students. The topic of today's lecture is about Drosophila embryogenesis, that is about Drosophila melanogaster. As you can visualize over your screen, the Drosophila melanogaster is commonly referred to as the queen of genetics. भाई यही Drosophila melanogaster को हम queen of genetics कहते हैं. इसे हम queen of genetics क्यों कहते हैं? Why it is called as the queen of genetics? And what are the major contributions about genetics that have been made through the Drosophila melanogaster study, and how it has been useful in the field of genetics in terms of illustrating the chromosomal map, the concept of linkage, crossing over, and then about the developmental biology, in what manner it has been used in developmental biology to understand the concept of evolution of life. Okay, so let us move forward in this lecture and look at the evolution of Drosophila melanogaster from a zygote into a complete fly. किस प्रकार से हम सबका एवोल्यूशन हुआ है, कैसे जीन्स मिलके किसी भी एक बॉडी के पार्ट को बनाते हैं, हम क्यों बायलेटरली सिमेट्रिकल हैं, why the right hand of our body resembles with the left hand of our body, why we are bilaterally symmetrical. All these answers are coming forward to you from the study of Drosophila melanogaster. So let us move forward. So Drosophila melanogaster has been used as a relatively very simple model for the process of cell differentiation, in what manner a single cell will form a multicellular organism like you and me. So how it has been produced? आप जैसा और मेरे जैसा multicellular organism कैसे बना है एक single cell से? इस चीज की स्टडी किसमें की गई है? ड्रोसोफिला melanogaster में की गई है. And so we would be looking at Drosophila melanogaster, that how this study has been performed. Okay, so that we are going to look forward. During fertilization, a single sperm cell enters a mature egg, and the haploid sperm and egg nuclei fuse to form a diploid nucleus. So during the process of fertilization, what we see is that a diploid nucleus is going to be produced. During the process of fertilization, the male sperm and the female egg fertilized in order to form a diploid Drosophila nucleus. Okay, so this diploid Drosophila nucleus will lead to the formation of zygote. And how does the division of this nucleus takes place in the case of zygote? किस प्रकार से डिप्लॉइड न्यूक्लियस का डिवीजन होता है zygote में? Let's see. Only a few hours after fertilization, the cells of a developing fruit fly embryo become irreversibly determined to differentiate into specific tissues in the adult fly. So just after few hours, the cells becomes completely differentiated in order to form the different components of the fruit fly. Okay, so those different components of the fruit fly will be formed after the differentiation of cell. एक बार जब एक जाइगोट का फॉर्मेशन होता है स्पर्म और एग के फ्यूजन से, उस जाइगोट के बाद में डिफरेंशिएशन होने लग जाता है. डिफरेंशिएशन ऑफ द सेल began to take place in order to form different parts of the fruit fly, such as the facets of the eyes, the forelimbs, the hind limbs, the antennapedia complex. All these different types of structures are being produced. We must view that how these structures are being produced. This exercise, you should understand the events of Drosophila embryogenesis and understand how the position of each nucleus determines its fate in the developing fruit fly. Okay, so we must understand the event of Drosophila melanogaster embryogenesis. How does the development of the embryo takes place? In what manner the embryo is going to be produced in the case of Drosophila melanogaster? That we have to understand. We have to completely understand the process of cell differentiation. That in what manner the cell is of the Drosophila melanogaster is going to get differentiated. किस प्रकार से embryo का development होगा एक adult fruit fly में, और किस प्रकार से प्रत्येक सेल हर एक सेल का डिफरेंशिएशन होता है कि किसी एक सेल को आई का सेल बनना है तो अलग बनेगा, किसी एक सेल को किसी और पार्ट का सेल बनना है तो अलग बनेगा. देन हाउ डज द डिफरेंशिएशन टेक्स प्लेस? यू मस्ट बी वंडरिंग that we have You must be wondering that we have 46 number of chromosomes present in our cells. हमारे अंदर 46 number of chromosomes होते हैं. अगर हमारे अंदर 46 number of chromosomes हैं, तब भी तो हमारे सेल्स जो हैं एक दूसरे से अलग अलग होते हैं. हमारा आई का सेल अलग है, पेनक्रियास का सेल अलग है, लीवर का सेल अलग है. तो ये सेल्स आपस में एक दूसरे से अलग अलग क्यों हो पाए? How the differentiation of these different cells has taken place, that we will have to understand. So moving forward, now we would be looking at how the differentiation of the cell is going to take place. After the haploid sperm and egg nucleus fuse together, the resulting zygote which is being produced undergoes n number of nuclear division. This is the zygote that is undergoing nuclear division. देखिए, एक बार जब zygote बना तो zygote के अंदर क्या हो जाएगा? न्यूक्लियस का डिवीजन होगा. The zygote nucleus is going to divide. As the zygote nucleus divides by 10 cleavage reactions, then how many number of nucleus will be produced? So the number of nuclei produced out of the 10 rapid cleavage divisions is 1024 number of nuclei which are going to be produced. So how many number of nuclei are going to be produced? 1024 number of nuclei. So these are the number of nuclei which are going to be produced. Okay, so when there is 1024 number of nuclei which are going to be produced, these nuclei are then going to migrate towards the periphery of the cell. Okay, these nuclei will slowly and steadily begin to migrate towards the periphery of the cell. ये nuclei क्या करेंगे? ये न्यूक्लियाई सेल की पेरिफेरी की तरफ मूव करने लग जाएंगे. वंस दे बिगैन टू मूव टुवर्ड्स द पेरिफेरी ऑफ द सेल, सेल के किनारे की तरफ मूव करने लग जाएंगे. अगर मान लीजिए ये एक सेल है, नो मैं हर्ट नहीं बना रहा हूं, मैं सेल बना रहा हूं. दिस इज नॉट हर्ट, दिस इज सेल. ओके, सो इट इज सिंसीशियम. नाउ द सेल्स विल बिगैन टू मूव टुवर्ड्स द पेरिफेरी. ओके, वंस इट बिगैन टू मूव टुवर्ड्स द पेरिफेरी, देन such type of condition in which there is a single cell but that cell is multinucleated is referred to as syncytium condition. बेटा एक ऐसी कंडीशन जिसमें सेल तो सिंगल सेल्ड है क्योंकि उसका सेल एक है लेकिन न्यूक्लियस बहुत ज्यादा हैं, इस कंडीशन को हम बोलते हैं syncytium. Such type of condition is commonly referred to as the syncytium, and this is the syncytial epithelium. This is the syncytial nucleus condition which is being produced during the embryogenesis of Drosophila melanogaster. After this syncytial condition is going to be produced, then the embryo is called a syncytium at this point because it is a single celled with multiple number of nuclei that are present. So in this particular case, the embryo is commonly referred to as a syncytium. Now after the formation of syncytium, what next is going to happen? एक बार syncytium बनने के बाद में फिर क्या होता है? एक बार यदि syncytium बन चुका है, then the nucleus will begin to migrate towards the periphery. Okay, they will begin to migrate towards the periphery. How? Look here. Once the division has taken place, then it is the migration of the nucleus that is going to start. Once the migration of nucleus starts, the nucleus will begin to migrate towards the periphery. After about 8 cleavage divisions, there are 256 nuclei which are being produced. They began to migrate towards the outer edge of the cell. जो 256 नंबर ऑफ न्यूक्लियाई हैं वो किनारे की तरफ चलने लग जाएंगे. ठीक है, एज दे बिगैन टू मूव टुवर्ड्स द आउटर पार्ट ऑफ द सेल, so they will lead to the formation of a structure that is commonly referred to as cortex. Okay, so this is the cortex region that is going to be appearing here. And in any structure, whether you would have talked about the root epithelium, root dissection, shoot dissection, you would have always heard about a structure which is commonly called as cortex. Another one is referred to as medulla. एक स्ट्रक्चर को हम कॉर्टेक्स बोलते हैं. बाहर की तरफ जो स्ट्रक्चर होता है उसे हम कॉर्टेक्स कहते हैं. अंदर की तरफ जो स्ट्रक्चर होता है उसे हम क्या कहते हैं? मेडुला. सो बोथ दीज स्ट्रक्चर्स कैन बी डिफरेंशिएटेड फ्रॉम ईच अदर ऑन द बेसिस ऑफ अरेंजमेंट ऑफ द न्यूक्लियस. in the arrangement of the nucleus. When it is towards the periphery, then it is called as the cortex. When it is towards the center, then it would be referred to as the towards the medulla region. Okay, so after the 8 cleavage division has occurred, 256 number of nuclei has been produced. Those nuclei have migrated towards the outer edge of the cell. ये 256 न्यूक्लियाई जब आउटर एज ऑफ द सेल की तरफ माइग्रेट कर गए हैं, then after this migration further 2 cleavage division takes place, leading to the formation of 1024 number of nuclei. How many number of nuclei will be produced? Approximately 1024 nuclei are going to be produced, and these nuclei are going to enter into the cortex region. Okay, so they are going to be entered into the cortex region of the developing embryo. Okay, most of them enters the cortical regions of the egg, and certain remains within the medulla region. अधिकतर तो कॉर्टेक्स रीजन में पहुंच जाते हैं, while some may remain in the medulla region. Okay, so there are certain number of nuclei which may remain in the medulla region. Okay, next point, clearing the drawing and moving to the next point. Now moving forward, we have to look that how does the further development takes place in Drosophila melanogaster. Now, after about 90 minutes of the process of fertilization के करीब 90 मिनट के बाद में, 90 मिनट ड्रोसोफिला के पास में है उसकी जिंदगी के सबसे इंपॉर्टेंट 90 मिनट. ओके, ये कोई नहीं उससे छीन सकता. इस 90 मिनट के बाद में ड्रोसोफिला के अंदर जब फर्टिलाइजेशन हो चुका है, then the majority of the nucleus have reached towards the cortex. Now majority of the nucleus have reached towards the cortex. There is no nucleus which is present in the medulla region. Okay, at this point the nucleus gains the ability to transcribe RNA polymerase. अब न्यूक्लियस के अंदर ये एबिलिटी आ गई है कि वो अपना आरएनए पॉलीमरेज ट्रांसक्राइब कर सकता है. दैट मींस इट हैज द एबिलिटी टू फॉर्म द एमआरएनए मॉलिक्यूल इनकोडिंग फॉर प्रोटीन्स. अब ये प्रोटीन्स को बना सकता है. बेटा जब ये प्रोटीन्स को बनाएगा तो प्रोटीन्स की वजह से इसका क्या हो जाएगा? डिफरेंशिएशन हो जाएगा. द ड्रोसोफिला सेल्स विल गेट डिफरेंशिएटेड बिकॉज ऑफ द फॉर्मेशन ऑफ प्रोटीन्स. ओके, सो हियर once it is able to form this RNA molecule, it has the ability to transcribe the RNA molecule, okay, RNA second genes, and then it will lead to the formation of proteins. इसके बाद में प्रोटीन को बनाना शुरू कर देगा. Once the protein molecules will be produced, then these proteins will be utilized in the cell. They undergo another 3 rounds of cleavage, leading to a dense packaging of 6000 column-shaped nuclei enclosing the central yolk. Then they undergo further division. इसके बाद में इनका फिर से division होगा, and as they undergoes division, they form 6000 column-shaped nuclei enclosing the central yolk. There is a central yolk which is going to be present, and there are column-shaped nuclei which are going to be present. यहाँ पर बीच में क्या हो जाएगा? At the center there would be a structure that would be referred to as yolk. यहां पर क्या बन जाएगा? यॉक बन जाएगा. ओके, एंड देन देयर वुड बी column shaped nuclei present here in the cortex. Column shaped nuclei, column shaped nuclei, okay, that would be present. Now further these nuclei have gained the ability to form the mRNA molecule. Now these nuclei will begin to perform the act of differentiation. अब इन न्यूक्लियाई के अंदर डिफरेंशिएशन होने लग जाएगा. दे विल बिगैन टू डिवाइड एंड फॉर्म स्पेसिफिक टाइप ऑफ स्ट्रक्चर्स. अब ये अलग अलग तरीके के स्ट्रक्चर्स को फॉर्मुलेट करना शुरू करेंगे. नाउ दे विल बी एबल टू प्रोड्यूस द different types of structure in the cell. Now how does they produce the different types of structures in the cell? That we will have to see. किस तरीके से कौन कौन से स्ट्रक्चर्स को बनाएंगे ये हमें यहाँ पर देखने की जरूरत है. So that we are going to look forward. Okay, so just deleting all the drawings. Moving forward, so you can find just after 2 to 3 hours of fertilization, cell membrane appears between the nuclei. Now, in this particular condition, a single cell has been divided into a multicellular embryo. अभी तक ये क्या था? एक single cell zygote था. zygote के बाद में आप लोगों को पता है zygote के बाद में क्या होता है? Cleavage cell division होता है. There is a cleavage cell division that is going to take place in the case of human embryogenesis. Here what is going to happen? The nucleus which have migrated towards the periphery, जो न्यूक्लियस किनारे की तरफ चले गए थे, उन न्यूक्लियस के अंदर सेलुलाइजेशन हो गया. Once they have gotten converted into particular cells, the embryo is getting transformed into a cellular blastoderm. There is a cellular blastoderm that is going to be produced from that particular embryo. That embryo has formed a blastoderm. So this is the cellular blastoderm. During a 1 hour period, from 2 to 3 hours after fertilization, cell membrane forms between the adjacent nuclei. There is the appearance of cell membrane. By 3 hours after fertilization, the embryo has been transformed into a cellular blastoderm. Now it would be referred to as blastoderm. That means this structure is now called as blastula. आप लोगों को पता होगा कि एक मोरुला होता था, उसके बाद में दूसरा हो जाता था क्या? Blastula हो जाता था. So how does this morula and blastula is going to be produced? That we have to look here. Firstly, it was the morula which was being produced. Now it has been converted into blastula. Okay. So then, or as all the cells have moved towards the periphery, जैसे कि सारे के सारे cells किनारे की तरफ चले गए थे, तो बेटा बीच में क्या रह जाएगा? बीच में there would be a hollow cavity that will remain. A hollow cavity will remain in the center, and this hollow cavity indicates the blastocele. ये बीच में जो cavity रह गया है, इसे हम क्या कह देते हैं? This cavity is commonly referred to as the blastocele cavity, which is going to be present. And this blastocele cavity indicates that this cavity is similar to the peritoneal cavity which is present in the case of human. हमारी body में भी abdomen में क्या होता है? Peritoneal cavity होता है. So this represents the peritoneal cavity present in our body. So this cavity is called as the peritoneal cavity. Okay, so that is peritoneal cavity, that is resembling directly the blastocele cavity. Okay, after 3 hours after fertilization, the embryo has been transformed into a cellular blastoderm, essentially a hollow ball of cell. So this is essentially a hollow ball of cell that has been produced. Moving forward, that how does the further development will take place? So see this. Now the nuclei have reached the edge of the cells. Still, the nuclei are totipotent, capable of differentiating into any other type of cells. What is totipotency? Totipotency refers to the ability of the cells to form any other type of cell. That condition is commonly referred to as totipotency. Totipotency होती क्या है? एक सेल की एबिलिटी कि वो किसी भी तरीके के सेल्स को बना सकता है, इसी को बेटा हम टोटिपोटेंसी कहते हैं. Now, these cells before cellularization were totipotent. That means if you are going to transfer one cell from one site to another site, अगर आप इस सेल को यहां से निकाल के यहां पर डाल देंगे या किसी और सेल को कहीं से निकाल के कहीं और ट्रांसप्लांट कर देंगे, then what is going to happen? Still a normal embryo is going to be produced even after the transformation has taken place, because the cells are highly totipotent. But once the cell has been differentiated to form a particular type of an cell type, then the transfer will lead to the absence of all the particular organs that these cells could produce. अगर मान लीजिए सेल डिफरेंशिएशन के बाद में मैंने इस सेल को डिलीट कर दिया, then this cell might be giving rise to wings. Okay, so those wings will become absent. These wings will become absent. These wings will become absent. तो ये जो विंग्स हैं ये एब्सेंट हो जाएंगे, ये नहीं बनेंगे, ये बन ही नहीं पाएंगे, खत्म हो जाएंगे. They would be completely finished. Okay, but before that, if the cells that could lead to the differentiated could be leading towards the differentiation to form wings, If those nucleus are being removed, if this supposedly if this nucleus removed, then what is going to happen? Nucleus removed, what is going to happen? Write in the chat box, let me see what do you think. When the nucleus will be removed, then what is going to happen? Okay, so I'm just taking this particular case. The nucleus has been removed, so what would happen? Kya hoga? Nucleus remove ho jayega, other nucleus will divide, other nucleus will divide, and a normal Drosophila melanogaster will be produced, and a Drosophila melanogaster will be produced, and this Drosophila melanogaster that would be produced will be completely normal. एकदम normal Drosophila melanogaster बनेगी, कोई भी change नहीं आने वाला. There would be no change that will be taking place. Okay, so is this clear to you all? I do think this would be clear. Let's move forward. Now, moving forward, what we get out of here is that the totipotent cells have been differentiated to form, the totipotent nucleus have been differentiated to form the cells. When the nucleus reaches the edge of the cell, they are totipotent, meaning they have not yet taken on a specific identity and can give rise to any cell type. Just after cellulization, however, the nucleus have been irreversibly determined, and once they have been irreversibly determined, they will lead to the formation of the blastomeres. Okay, however, the nuclei have been irreversibly determined to differentiate into specific tissues and to the adult fruit fly. The location of each nucleus is determined a determining component for its phase. Okay, so where the nucleus is going to be present, it is going to be determining its fate. देखिए किसी भी न्यूक्लियस का क्या बनेगा, वो न्यूक्लियस एंटीरियर पार्ट को सेल्स को बनाएगा या पोस्टीरियर पार्ट के, ये किस बात के ऊपर डिपेंड करेगा? इट विल डिपेंड अपॉन द एनवायरमेंट ऑफ दैट पर्टिकुलर न्यूक्लियस. सी इट इज वेरी क्लियर. So whatever is going to be produced, it is based upon the environment that that cell is going to receive. Okay, so if a cell has to form the anterior component, then it must receive certain specific type of signals to form the anterior component, and if the cell has to form the posterior part, it must receive certain other type of the signal to form the posterior part. Okay, so cell को anterior part बनाना है तो अलग signal, cell को posterior part बनाना है तो अलग signal. सर क्या होगा अगर मैं दो अलग अलग प्रोटीन, एक ही प्रोटीन को, एंटीरियर वाली प्रोटीन को एंटीरियर पार्ट में भी दे दूं और पोस्टीरियर पार्ट में भी दे दूं, तो क्या होगा? Supposingly, sir, what is going to happen if I am going to insert a important anterior part protein in the anterior as well as posterior part? Can you write it out? What will happen? क्या होगा? Then the Drosophila will be produced with two heads, okay, and with no posterior part. तो ऐसी ड्रोसोफिला बन जाएगी जिसके दो सिर होंगे लेकिन कोई posterior part नहीं होगा. Such type of Drosophila could be produced. Okay, so it is all on the basis of the signaling sequences or the protein signals that helps in the differentiation of the Drosophila melanogaster. Okay, so this is how the differentiation actually takes place in the cell. Next point. So let us move forward and look at how the further division is going to take place. Now, what we find here is that further the different sides of the Drosophila are going to be determined. The anterior side, the posterior side, the dorsal side, and the ventral side. Okay, so these are the four different sides which are going to be firstly determined in the case of Drosophila before the further evolution is going to take place. इससे पहले कि ड्रोसोफिला का आगे कुछ evolution हो, सबसे पहले ये चारों साइड पता कर दी जाती हैं. एंटीरियर कहाँ पर है, पोस्टीरियर कहाँ पर है, डोर्सल कहाँ पर है, वेंट्रल कहाँ पर है. Once they are being decided, then the further division or the differentiation of the cell will take place. Moving forward. Now, first type of genes that we are going to talk is about the maternal effect genes. Now, what are these maternal effect genes? Maternal effect genes means the genes whose product are present within the cytosol of the egg, and they are the one which will determine the fate of the egg. मैटरनल इफेक्ट जीन्स बेटा ऐसे जीन्स होते हैं जिनका प्रोडक्ट साइटोसोल में होता है, और इनका प्रोडक्ट जो साइटोसोल में है वो डिटरमाइन करता है दैट how the differentiation of the cells is going to take place, in what manner the cells are going to be differentiated. Look here. Now, there is an mRNA which is going to be present towards the posterior part of the egg, and this mRNA is commonly referred to as the Oscar mRNA. The Oscar mRNA encodes an RNA binding protein that is responsible for the assembly of polar granules, and these polar granules will give rise to the gametic cells in the case of Drosophila melanogaster. अब देखिए यहां पर कुछ mRNA हमेशा ही एक्सप्रेस होते हैं किसके अंदर, इन द एग ऑफ ड्रोसोफिला, before even the process of fertilization can take place. There is an mRNA concentration that is found in the egg of the Drosophila melanogaster. All such mRNAs are commonly referred to as maternal effect genes. Those mRNA will be referred to as maternal effect genes. Their concentration will be found in the entire sequence of the Drosophila melanogaster. Okay, so their entire sequence will be present in the entire concentration, or their concentrations will be present towards the anterior, towards the entire sequence of the Drosophila melanogaster. So there we are going to form the different types of molecules will be there. Okay, so we'll be looking at all these different types of molecules and their concentration in this particular topic. There would be a protein that would be referred to as bicoid protein. The accumulation of bicoid protein will take place towards the anterior position, and caudal will take place towards the posterior position. Okay, so all these mRNAs will lead to the differentiation of the Drosophila embryo. Just let us have a look at all these mRNA. See this. Oscar mRNA will give rise to polar granules even before the nucleus has divided. The Oscar mRNA was previously present. Oscar mRNA has been converted into proteins. Proteins have togetherly with the Oscar mRNA bounded together to form polar granules. Then, as the nucleus will migrate towards the polar granules, they will lead to the formation of gametic cells. Approximately 30 nucleus that migrates into the polar plasm buds off from the main body of the embryo along with polar granules, and the resulting pole cells differentiate into the germ cells. These resulting polar granules will combine with the nucleus, will get differentiated into the germ cells. They will get differentiated to form the germ cells. Recording. So as you know that the cell fate is always being determined by the localized mRNA molecules which are going to be present. Okay, so cell fate is being determined by the localized mRNA molecule. You can find this is the fertilized egg. The eggs that migrated towards the posterior side will get transformed into the gametic cells. Okay, so these nuclei just migrated towards the posterior side and will get transformed into the germ-line cells. Which cells they will form? They will form specifically the germ-line cells. So I'm just pointing out the germ-line cells. Sorry for the disturbance. Okay, pointing out the germ-line cells here. These are the germ-line cells that are going to be produced. Okay, moving forward after this, then this is the poroplasm, and they will give rise to the germline cells. These are the germline cells that have been produced. So what has happened? It is these nucleus that have migrated towards the posterior part and have given rise to the pole cells, whereas those nucleus that migrated towards the anterior part will have given rise to the head region. As it has migrated towards the anterior part, so there would be a head region that is going to be produced. क्योंकि एंटीरियर पार्ट की तरफ चला गया तो हेड बन गया, पोस्टीरियर पार्ट की तरफ चला गया तो पोल सेल्स गैमीट्स बन गए, डॉर्सल की तरफ चला गया तो विंग्स बन गए, वेंट्रल की तरफ चला गया तो हाइंड विंग्स बन गए, फोर्ड विंग्स बन means that will lead to the formation of three paired appendages or the halters that would be produced. So all these types of structures could be produced from the Drosophila melanogaster. Okay. Now, further moving forward, what we are going to find is that how the cell fate is going to be determined. So, let me show you to you that how the cell fate is going to be determined. The cell fate is being determined by the concentration of several proteins, which are although present in the cytoplasm even before fertilization. Fertilization होने से पहले ही कुछ प्रोटीन्स होती हैं जो साइटोप्लाज्म में होती हैं और उनका कंसंट्रेशन ही सेल का फेट डिसाइड करता है. विच आर द प्रोटीन्स दैट आर प्रेजेंट इन द साइटोप्लाज्म? द प्रोटीन दैट इज प्रेजेंट इन द साइटोप्लाज्म हियर are the maternal effect gene proteins. Okay, then the sperm enters, the fertilization takes place. Okay, and it is this fertilization that lead to the formation of large number of nucleus that began to migrate towards the periphery. Now the concentration gradient of several different proteins will help in the redistribution of the dorsal and the ventral polarity in the case of Drosophila melanogaster. Now, what is this spasal protein? How does the dorsal and ventral polarity is going to be produced? That we are going to look in our next class tomorrow evening at the same time. Okay, so we are going to just finish it out here only. This is just an introductory lecture about Drosophila melanogaster. In tomorrow's lecture, we would be looking at the maternal effect genes and how the anterior posterior, the dorsal and ventral polarity of the Drosophila melanogaster is going to be produced. किस प्रकार से अलग अलग प्रोटीन्स ड्रोसोफिला के अंदर एंटीरियर पोस्टीरियर और डॉर्सल और वेंट्रल पोलैरिटी को प्रोड्यूस करती हैं, इन सभी चीजों को हम लोग कल के लेक्चर में एक बार अंडरस्टैंड करेंगे और इसे देखेंगे. So I will meet you tomorrow at the same time. That's all for today. Hope you have enjoyed the lecture. Thank you.